Now, just to sort of throw a wrench in the works. Remember, back in the dark ages of like last October, for those of you that took Biochem 1 last semester, maybe even late September, back in '63. Another song lyric reference. We said that arginine was one of the essential amino acids. And if we say it is essential, what does that mean? We don't make it. Did I lie to you? Well, we made it, but what's the problem with this? We've got to go back to make our starting material to complete the cycle, which means we can't use this, we can't pull this out to use it for making proteins without depleting our urea cycle intermediates, which then depletes our ability to handle the nitrogen coming from other amino acids that we eat. So we make it as an intermediate in the cycle. From that last question on that last test, what is the role of all these intermediates in the cycle? Catalytic. They're catalytic. So if we remove our catalyst, we remove our capacity for doing this. So that's why we can, without really lying to you, make arginine in the urea cycle as an intermediate, but because we have to use it immediately, that doesn't mean we're making net arginine that we can use as our source of arginine for building proteins long term. Does that make sense? But it does mean that if we were undergoing starvation and we had used up some of these intermediates, how do we replenish our urea cycle intermediates?
Break down proteins.
Break down proteins. You eat some protein that has arginine, that arginine can immediately replenish that urea cycle intermediate. Right? Okay, so now we've got arginine here. We're going to hydrolyze that. That is, water is going to be the nucleophile. We're going to hydrolyze that sort of in the reverse of this reaction. You know, where nitrogen attacked here and water left. Water is going to attack here, but instead we're going to break this bond. We make urea out of the two new nitrogens in the carbon, and the ornithine is back. Ready to start the cycle again.
So this will just keep going as long as you add the carbon.
As long as you've got nitrogen coming in and ATP, as long as you can feed in carbamyl phosphate.
So, like with the citric acid cycle, we used, it wasn't what we brought in that was being formed. Like, does that make sense? It wasn't. No.
let's try to word that.
It wasn't the carbons from the acetyl-CoA that became the carbon dioxide.
Right. The first cycle through.
Right.
Like eventually, if you followed it, it would come out.
In this one,
it is the nitrogens that we're bringing in.
So
that ultimately are forming the urea.
So the intermediates always stay. They don't ever come out.
Right. So if we radioactively labeled this nitrogen in ornithine, our urea would not be radioactively labeled. Yes.
So you need a aspartate too?
Yes. Right? So you got one nitrogen coming in as amino. The other nitrogen is coming in from aspartate.
Where does the ATP use that step?
In which step?
The arginine substitute.
Well, where would we guess? I'm going to ask you that. What are we usually using ATP for in a reaction like that?
Like on the enzymes that actually part of it? Or?
It's a nucleophile?
No. What do we usually, in those kinds of reactions, what do we use ATP for? I mean, what do we use it for up here? To make a bond that we could do a favorable substitution. So I would guess that an intermediate probably is maybe a phosphorylated nitrogen there that we can use to make something, maybe phosphorylation there. I don't know what the intermediate is. But we're creating a bond where we can do a favorable substitution.
Okay. You
Right?
just said that the nitrogen just attacks the carbon.
Yeah.
Well, it's not
To
like-
make it energetically favorable, the ATP's involved. I don't know the mechanism of that. Okay? But again, right, if we're talking about energy, right, here's 2 ATPs worth of energy. This ATP, since we're making pyrophosphate, that's going to be hydrolyzed by the inorganic pyrophosphatase. So that's another 2 ATPs worth of energy. So for each mole of urea that we make, we require at least 4 moles of ATP energy. Right? For every mole of, no, and since that's 2 moles of nitrogen, for every 2 moles of nitrogen we excrete, we have to use 4 moles of ATP energy to create the urea. So what does that tell us? Must be important. If we're going to invest that much energy, it must be important. Right? And then the fumarate can be converted to malate. The malate can be converted to oxaloacetate, and I'm going to differ here. I'm not going to take it to gluconeogenesis. What am I going to do with that oxaloacetate? Melanie?
Citric acid carbon.
No?
Oh, you're going to make the alpha keto.
No. I'm going to use that oxaloacetate, I'm going to transaminate it to make the aspartate again. Right? So it's going to react with glutamate to make that aspartate. Right? So you've got this cycle, glutamate reacting with, alpha-ketoglutarate reacting with other amino acids to make glutamate. Some of our glutamate will go this way, right, to release ammonia as a result of the alpha-ketoglutarate dehydrogenase reaction. Some of our oxaloacetate will react with glutamate in a transamination reaction to remake aspartate that we need for that step of the reaction. So this is a time when I'm going to disagree with what the book says. Yes, it could go that way, but probably under the conditions when we've got this high nitrogen load, you're going to have 2 cyclic shuttles, 2 ways of getting nitrogen from glutamate into the urea cycle.
You said that the carbonyl phosphate, when we were making that, it was in the mitochondria. Is the rest of it in the...
Yeah, right. These 2 yellow circles, what do they represent here? Membrane transport, right? So the first step, ornithine to citrulline, occurs within the mitochondria. Citrulline can be transported out. The other steps occur in the cytoplasm. So you get back to ornithine, it's transferred back in.
Is that in the mitochondrium? Is that in the intermembrane space or the matrix?
The matrix. Right? Because remember, we wouldn't have to worry about this transport in the intermembrane space. Right? Except, anything smaller than proteins can get through the outer membrane. So we can transport that in. So why would we do this in the mitochondrial matrix? What's different about the mitochondrial matrix versus the cytoplasm?
pH?
Yeah. There's a lot of ATP in there. So we can get this started in the place where we have the greatest local concentration of ATP. Okay?
Where did you say we get our the carboxyl phosphate? The carbon.
This?
Yes.
It's carbon dioxide in the form of a bicarbonate. Right? So we've always got carbon dioxide around. Okay? Any questions about the urea cycle in our brief overview? Yeah, Robbie?
How'd you say the fumarate left?
It's an elimination reaction, right? If we're thinking about mechanism, we've got this carbon adjacent to a carbonyl, right? So we could deprotonate that, making that carbon negative. All we need is a leaving group. If we protonate that nitrogen, right? Our arginine can act as a leaving group, leaving behind fumarate in a reverse Michael type elimination. Which aren't we starting to be able to see those? We look and we go, oh yeah, I can see where that would go. This is why one of my ideas that I probably won't ever get around to is to write an organic chemistry book for biology majors that focuses only on the organic chemistry that biological systems use, focuses on it from that perspective. But there are no books out there. I'd have to write my own book. I don't know if I'm that disciplined. As a matter of fact, I'm pretty sure I'm not that disciplined. But,
Maybe when James.
yeah, maybe when I'm not spending 7 hours every Saturday at a baseball field, it will help. But are there any questions? All right, there are some mechanism things that we just aren't going to have time to talk about. Metabolism of amino acids, that's fun. There's all kinds of fun stuff we haven't gotten to do yet.
Oh, looks like you
Easy
need
guys.
Next semester.
Yeah.
I could.